Thank you, Brian, and I really appreciate the invitation to speak. I'm going to do it differently. You know, we often, as Dr. Heist mentioned, we kind of sit back and let our students and residents do the research, and we come and present their work. I decided to do it differently this year. I've actually brought the students and residents that did the work to present it to you. So I will introduce it, and this is Joyce Quake. She's a 6-year neurosurgery resident. She only has 15 years to go to finish residency, and Jamila is hoping to go to med school next year. So these are preliminary results of an ASAP-funded research that we've been doing. Joyce?
Good morning, everyone. I'm Joyce. Thank you for having us here. As most of you know, Chiari malformation is a congenital disorder, and it can be inherited in up to 10% of cases. And we do have evidence of that from twin studies and other multiplex family studies. The literature has over 30 publications, including 250 familial Chiari malformation. There are also studies that look at syndromes, and Chiari has been associated with many syndromes, most commonly craniosynostosis, neurofibromatosis, Epiphyseal syndrome, as well as others. And the literature also has individual case reports or genetic studies that look at genes that may explain Chiari malformation and in specific familial Chiari malformations. However, so far there has been no evidence of inheritance, and not one gene mutation can explain a Chiari malformation. So a disease or inheritance can be done through mutation when in the DNA one of the nucleotides is deleted, for example, and then this is inherited permanently. Another form of inheritance is through epigenetics, and this is through DNA methylation. When one of the nucleotides, in specific the cytosine, the C nucleotide, gets a methyl group attached to it, and when that happens, the gene expression changes and the function of the gene changes, but this can be reversible. It's not a permanent effect. So we propose to look at familial Chiari I malformation through and study their epigenetic mechanism, that is the DNA methylation, because DNA methylation is the most studied epigenetic modification that can change the gene expression. And once we identified these patients, we wanted to do a whole genome methylation, that is to look at all these regions that have the methyl group and compare them between familiar Chiari patients versus the controls. We wanted to have 30 Chiari patients at the University of Wisconsin in Madison, and familial Chiari is defined as more than one member of the family who is diagnosed with Chiari malformation. However, controls are patients who have no evidence of Chiari or other neurologic pathologies on their MRIs. The population age ranged between 15 and 45 years old, and these were age and sex matched. We decided to collect saliva because it's the easiest and has the minimal risk of collection, and studies have shown that saliva is as good as, if not superior to blood, when looking at epigenetic changes in the brain. Once we got this data, we did the whole genome methylation sequencing, that is identifying every methyl group that has been attached to the cytosine. We've identified 30 patients so far and have enrolled 22 patients from 13 families in our study. We have 6 males and 16 females. The age at diagnosis ranges between a year and 43 years with a mean of 17 5.7 years. The mean tonsillar descent was 8.3 millimeters. The most common sign and symptoms of presentation of the Chiari patients was headaches, neck pain, some hydrocephalus, hearing loss or tinnitus, Syringomyelia, eye problems, especially nystagmus, and scoliosis.
And then, as Joyce has mentioned, very commonly we see multisystemic effects present with Chiari I malformations. And so our next step with our cohort was to better characterize that presentation. So we sent out a self-report survey to review 10 systems, which we'll go over more in depth. But briefly here in red is the Chiari group, and in blue we have our controls. Overall, we can kind of see that the Chiari I group tends to report more system involvement, and we'll dive into each of these a little bit deeper here in a moment. So starting off with kind of our general category, this was more of a catch-all. We saw some overlap with EDS, multiple sclerosis, as well as one instance of congenital fusion of the cervical spine. We also saw pretty drastic vision involvement in our Chiari group, typically presenting as blurred vision, also including involuntary shaking of the eyes and vision changes, as Joyce has mentioned, a common presentation with CM1. With the hearing review of systems, we found that 32% of our CM1 group presented with tinnitus or ringing of the ears. And then with our musculoskeletal review of systems, we saw a big proportion of our group present with neck pain as well as flexibility of the joint, and pretty equal report for fractures between our controls and our CM1 group. With the skin group, we saw 7 individuals report skin findings or birthmarks. And then neurologically, as Joyce has mentioned, we saw a significant portion of people reporting headaches and neck pain. Again, common things that we would expect with the CM1 presentation. And so the next part of our characterization of this group of 22 was to better understand any previous genetic testing that has been completed on these individuals. And so first, to kind of take a step back, we wanted to kind of recognize what has been known thus far about CM1 and genetic testing. So as Joyce has mentioned, there's no single gene that's been previously identified despite this known and recognized inheritance of CM1 transgenerationally. Despite this, we have seen some other syndromes, as some of the ones that are mentioned here, that tend to be associated with this clustering. And so we'll actually go over a patient today with this FSCB gene. And again, these are syndromes that are associated with CM1, but don't explain the clustering that is presenting in some of these families. And so within our group of 22, we captured 13 families, 6 of which we had more than one individual enrolled for. So we had 2 or more individuals enrolled. In 7 of the families, we had Chiari I malformations in the same generation. There were 4 sets of siblings captured, 2 sets of twins, unknown zygosity, meaning we are unsure molecularly if they're fraternal or identical. We had 1 set of first cousins, so 2 maternal first cousins. And then we had 6 families with different generations, so 5 instances of father-to-child transmission, and then 5 instances of mother-to-child transmission. And then just to kind of focus in on the twins in particular, since those are a great example and area to focus on for understanding genetic and epigenetic inheritance and just focusing research efforts. We had 2 sets of concordant twins, so concordant meaning they were the same sex, and then we had 1 affected triplet pregnancy. 2 of those siblings are deceased, and we did have another affected sibling, and so that was captured within our group. And so the previous genetic testing that we found through our report were 7 individuals that were actually evaluated by our genetics clinic. Of those 7 individuals, 5 of them were not recommended to have any genetic testing. So that was either they did not meet criteria for genetic testing or we did not have a recognized gene at that time. And so that left 2 individuals that ultimately pursued genetic testing. That testing revealed for our first individual, the testing was whole exome sequencing and whole genome sequencing. We're actually going to go through that in just a moment, so we'll put a pin in that. But participant 13, as you can see here, Fragile X and microarray testing, that was completed for some developmental concerns, and that ultimately came back negative. So no known explanation genetically for their Chiari I malformation or other presenting concerns. So participant 22, this individual was a 20-year-old female. She presented at birth with congenital cervical fusion of the spine. She had subsequent scoliosis and generalized hypermobility. She went on to develop headaches as well as some tics, which is why she was ultimately referred to our genetics clinic. At that time, whole exome sequencing revealed a variant of uncertain significance. So an inconclusive genetic testing result for Tourette's syndrome. She was ultimately reanalyzed with whole genome sequencing a few years later. That identified a variant of uncertain significance in a maternally inherited Camerati-Engelmann syndrome, which is a bone dysplasia condition. And so we'll take a look at her pedigree here in just a moment as well. So as we can see here, just briefly, I guess, take it a step back, the pedigree here in the boxes, those are representing males. The circles are going to represent females, and then the slash across any given symbol is going to just represent that they've passed. So looking here at the very bottom, we have on the right shaded our participant 22. So that individual has Chiari I malformation as indicated by red. Her sister was also found to have Chiari I malformation and actually underwent 2 surgical decompressions. Her sister also presented with Syringomyelia, and both her sister and her mother had a skew foot.
Okay, to get into the actual study, the molecular study that we've done, as Joyce mentioned, we captured saliva from these 22 patients as well as the controls, and this is what we found. And I will try to explain some of these graphs. Some of them can be confusing. The bottom line is that when we look at global DNA methylation, there was no difference between the control and the Chiari malformation. That tells you that there is not some major disconnect between the 2 groups. These are homogeneous groups. That's a good thing to start with. But when we looked at every single cytosine in their DNA to see if it's methylated or not, we saw a lot of changes. So this middle plot is called a circus plot. The outside shows you all the chromosomes. So 22 chromosomes, we excluded the X and Y chromosomes. In the inside, the red circle is every cytosine that's hypermethylated, has more methylation, and the blue one is every cytosine that's hypomethylated, has less methylation. And we find that there are over 2,000, 2,500 methylation changes that separate the Chiari, familial Chiari group from the control group. And then, and these are all associated with genes of various a function on the right. Now, when we plotted them a little bit differently to see where these methylation changes are, 3 families stood out. So you can see the one in yellow, see which is the one in yellow here, family 11, family 9, and family 5. We're going to concentrate on 5 and 11 because they really were very, very separate from the cohort. And when we looked at each one of these families, we found that there are significant methylation changes between them and between them and the average cohort. But also we found that there are some genes, some methylation sites that are that overlap. They're similar in some ways. So when we looked at them further, we found again that there are thousands of genes that are identifiable that separate these from the rest of the cohort in both families. In fact, also the third family that we're not talking about here. And then a lot of these genes have something to do with neurological function. So, that's actually quite good to have found something that separates three distinct families from the rest of the cohort. That means that there are epigenetic functions at stake here.
And then kind of returning to our pedigree and review of systems in conjunction with those epigenetic findings, helpful to characterize what was similar presentations between these individuals and what differed. For family 5, we did find that all 3 of these individuals reported headaches and vision involvement. Interestingly, the brother in this case was asymptomatic. He was not found to have his Chiari malformation until after the sisters were diagnosed. The 2 twins both were found to report some blurred vision. Our individual on the right here did have scoliosis. And then a major difference for the other 2 was reporting some ringing of the ears. But again, just one generation affected here. We didn't see transgenerational patterns. And then family 9, so this again was the family off to the left of the plot that we just saw. Family 9 includes participant 22, so this should look a little bit familiar. So again, we have the 2 individuals, one generation, both affected with Chiari I, and we had those 2 variants of uncertain significance. So no known genetic finding here in the coding, but an interesting epigenetic feature. And then finally, family 11. All of these 2 twins were both affected and both were reporting headaches. They also interestingly reported bone fractures and some skin findings. They differed in their presentation in that one of them had Syringomyelia and the other did not. The individual with the Syringomyelia did undergo bone decompression.
Okay, so what does it all mean? For years, we've looked at inheritance assuming that there are mutations, which is causing one family to have a particular disease process. Now we're understanding there is way more than this, that you can change the function of a gene without having a mutation, without having a DNA sequence variation or an error in that sequence. And that's what epigenetics is. And I'm going to explain it to you in words that we all understand, because it's a difficult concept. Imagine the piano. There are 88 piano keys. These are your genes. Okay? Now you can imagine that as humans, we have 25,000 genes, which is the same exact number of genes that our animal counterparts have. And yet we're very different. Okay? Each one of our cells has an identical set of genes than the other. So the eye has an identical set of genes than the foot, and yet they're different. The difference is epigenetics, is how these genes are being played. So you have 88 keys, but billions of songs. Every time you press a key, you've activated a gene or deactivated a gene in some way. I'm going to give you a better example even that one of my other students in the lab came up with, I thought was brilliant. Pandora music. Most of you, I assume, know Pandora music. So all of the Pandora radio is your entire DNA. That's the code that you have. The stations are the genes that we use. And every song is one gene. So what do? You play a song. If you like it, you tag it. If you don't like it, you untag it. This is what epigenetics is. What you're telling your DNA, activate this gene or deactivate this gene. So what is it that activates and deactivates the genes? The environment. So epigenetics is set up to deal with environmental changes. We can't start out with the same exact gene expression pattern in babies and have the same one when we're 80. It just doesn't make sense. We have to adapt to the environment, and epigenetics are the way to adapt to the environment, which means it involves everything that we do, including diseases, and especially inherited diseases, because even though a lot of these methyl tags or epigenetic marks that you accumulate over the years get presumably erased in the next generation, some of them persist, and you inherit something. So what we're seeing here is a disease process, Chiari I malformation, that is inherited in at least 10% of patients, and yet no mutation has ever been identified to consistently predict this or explain it. So epigenetics has to be at play. There haven't been any studies on this. This is really, ASAP has funded one of the very first studies to do this, and we're very excited about our preliminary data and hope to expand them further. The other thing I would add is there is an environmental component to epigenetics. And the question is, how much is there an interaction between environment and predisposition to have Chiari malformation? That's something that we don't know, and we would love to study. So here we're showing that DNA methylation signatures in saliva distinguish between familial Chiari malformation and control subjects. That these DNA methylation changes are particularly prominent in probably 3 families that we've identified. They overlap between 2 of the families and are primarily tied to synaptic and other neuronal functions. So it's very appropriate to the Chiari malformation. So we would love to replicate and validate these results with larger cohorts now that we have really good preliminary data. We would like to profile earlier ages before you accumulate a lot of methyl tags over the years. Here our lowest age limit has been 15 because, for practical reasons. And we would like to study other epigenetic mechanisms. DNA methylation is not the only one. The histone modifications, microRNAs, and there are others. So I'd like to thank the research team, including Jamila and Joyce, our funding agency, ASAP. Thank you again, and we'll take any questions at the break. Thank you.